Okay. Hello, everyone. This is Srikanth Bollam, Associate Scientist in FP2A genotyping team of Tigers at ICRISAT. So today I'll be presenting the molecular perspective to crop improvement in terms of sorghum and pearl millet. So our ultimate aim of our work is to improve the water use and yield stability in sorghum and pearl millet. So in order to achieve that, we aim to explore the natural variation for water use efficiency and yield-related traits in global diversity germplasm panels of both sorghum and pearl millet. So here for sorghum, we are using sorghum mini coset, which comprises of 240 accessories and which represents all the 5 basic and 10 intermediate races of sorghum. So for pearl millet, we use PMI gap set, that is Pearl Millet Inbred Germplasm Association panel, which comprises of 345 accessions and which represents the genetic diversity of major pearl millet growing countries, that is 27 countries. So screening of sorghum and pearl millet accessions for water use and crop production related traits is carried out at ICRISAT by our phenotyping team using lysi field phenotyping platforms. So for sorghum, 2 seasons data is available. For pearl millet, also 2 seasons data is available. So with the available 2 seasons phenotyping data, and also we have a high quality informative SNPs for this sorghum mini core set. We have generated HabMap from this high quality informative SNPs. That is approximately 18,000 SNPs we have for this panel. So by using both phenotypic and genotypic data, we have done GWAS analysis, and which resulted in a significant market rate associations for key traits like total biomass, transpiration efficiency, water use efficiency, water extraction, and grain yield related traits. And also we have done structure analysis, which resulted 8 major clusters for this panel. So in order to deep dive into the molecular and molecular networks which underpinning this imported trait, we have done some transcriptome analysis also. So we have used contrasting sorghum genotypes, which we have narrowed down from the pooled data, 2 seasons pooled data, and we have used these genotypes for dry down experiment. So we have conducted a glasshouse experiment in at ICRISAT, and these contrasting sorghum genotypes were subjected to drought stress at vegetative stage, that is approximately 21 days stage. And we have collected shoot and root tissues at different time points from both the well water and water stress conditions. So these samples were subjected to transcriptome analysis. And also we have designed from several gene-specific markers for QRT-PCR analysis. So here you can see the differentially expressed genes, top 20 up and down regulated genes in contrasting sorghum genotypes under varying water conditions. These are shoot tissue samples. Here you can see the differentially expressed genes in root tissues also under varying water conditions, in same genotypes. So now I'll start pearl millet genotyping activities at ICRISAT. So as I mentioned, we are using PMI gap set. We have shared the seed samples to for lysometric study. So we got first season data last year and second season data just 1 week before. So I have worked with first season data. Based on first season data only, we have narrowed down, we have continued our work. So we have whole genome resequencing data with more than 29 million called SNPs. From that pool of data, we could able to narrow down high quality informative SNPs to 67,000 SNPs. So with this genotypic information, we move to GWAS analysis. Here you can clearly see the density and distribution of SNPs across the chromosomes in PMI gap set. Yeah, pearl millet GWAS work is conducted based on first season data, as I mentioned. Initial analysis revealed significant market rate associations for key traits such as total biomass, transpiration efficiency, water use efficiency, grain yield, water extraction, etc. So we also generated some gene-specific primers and synthesized these primers for QRT-PCR work. Similar to pearl millet genotyping activity, we also conducted a dry down experiment for deep diving into a set of contrasting pearl millet accessions to study the molecular networks which are underpinning this water use efficiency trait. So based on first season data, we have identified contrasting pearl millet accessions. So we have chosen two better performing accessions and one poor performing accession, and we have subjected to drought stress at vegetative stage. So we have collected shoot and root tissues at vegetative stage. At booting stage, we have collected flag leaf, root, and panicle. And at maturity stage, we have collected flag leaf, root, and panicle samples under both the conditions. So these sampling completed the last 15 days back itself. So we are ready for transcriptome analysis. We'll be giving these samples for transcriptome analysis. So future works include validation and integration of genes identified through GWAS and transcriptome analysis in sorghum. So, and also the publication of the genotyping activities in sorghum. And also GWAS analysis with the second season and pool data analysis is pending as we received the data last week itself. So I'll be completing that. And also transcriptome analysis is our priority now. And also manuscript on pearl millet genotyping is one which is very important. And also differential expression of WE, water use efficiency associated genes in contrasting sorghum and pearl millet accessions under varying and water use efficiency conditions. So in this last 2 years, we could be able to publish different publications in different journals. So these are the publications. Okay, my sincere acknowledgements for all the partners in our program. Thank you.